We're going to look at the most common human blood group, the ABO blood group. We'll be looking at facts and disorders. In the United States, the most important blood group is the ABO group. This group has three alleles. They are the A, B, and O. An allele is a gene. A person can only inherit two alleles or two genes for this trait. Take a look at the chart on the right side. You can see some of the possibilities in blood types for offspring with different blood types for parents. You should be able to determine the phenotype, that is the blood type of a person, if you know their genotype. If their genotype is two A's, their phenotype is A. If their genotype is AO, again, their phenotype is A because A is dominant over O. If their genotype is two B's, their blood type is B. If their genotype is BO, their blood type again is B. That's because the B gene or the B allele is dominant over O. If a person's genotype is AB, their phenotype is AB because A and B are codominant, so both are expressed when they're present. And if their genotype is two O's, their phenotype is O. As you look over the last slide, you should be able to see that A and B are codominant. That means when both are present, both are expressed. In addition, A and B are both dominant over O. So that means that when A or B are present, they will be expressed and not O. In order to understand blood and blood diseases, you need to know what an antigen is. An antigen is any molecule that can be recognized by your immune system. In red blood cells, the antigens are actually glycoproteins located on the surface of the red blood cell. In the diagram on the right, type A you can see has green triangles on the surface. That is the antigen. In type B, you'll see blue circles. Again, that is the antigen. Type AB would have both the green triangles and the blue circles. I want to be sure that you understand what an antigen is. If you look at the red blood cells that are here, the one on the left, the one that's type O, has no antigens on the surface, no proteins on the surface. That labeled blood type A has triangular blue antigens on the surface. Type B has antigens, but they have a different shape and they are a different protein. And type AB contains both the antigens in type A and the antigens in type B, and therefore are type AB. We're going to look next at some examples of antigens and blood types. Look at this red blood cell. On its surface you can see the A antigen represented by a square, the B antigen represented by a triangle, and the Rh antigen represented by a notched square. The Rh factor is just another protein that can be found on the surface of some red blood cells. We're going to talk more about the Rh factor later on in the lecture. On the left is the red blood cell we saw in the last slide. Look at the two cells on the right side and see if you can figure out why one is A positive and one is A negative. Again, the original red blood cell is on the left. Compare that to the two red blood cells on the right and see if you can determine why one is B positive and one is B negative. The two cells on the right are AB positive and AB negative. Is that what you would have predicted? Does it make sense to you that the two cells on the right are O positive and O negative in comparison to our model on the left? We've talked about antigens. Let's talk now about antibodies. Antibodies are specific proteins that are produced by your immune system. They bind to the corresponding antigen. People who have a particular antigen do not have the corresponding antibody, or they would clump together. What they do have is antibodies to any red blood cell protein they do not have. To see this reaction of the antibody and antigen, put this slide in presentation mode, and you should see no reaction in the Rh negative blood, but a clumping or clotting in the Rh positive blood. Put this slide in slide mode and look at the right side to see where clumping is occurring. See if you can determine what the blood type is before checking out the answer on the left side of the slide. A blood type is determined by the antigen present on the surface of the red blood cell. So blood type A has antigen A, blood type B, antigen B, blood type AB, antigen AB, and blood type O has no antigen. See if you can figure out what the antibody for each of these would be. Remember, you don't want an antibody to an antigen that's present. Here are the frequencies of these 4 blood types in the United States. Type A is 41%, type B is 10%, type AB is 4% and is the least common, and type O is 45% and is the most common. You've probably heard the terms universal donor and universal acceptor. The universal donor is the blood type that can be given to all other blood types, so that means that it can't contain any protein that might elicit an immune response in the individual to whom the blood is given. Type O- is the universal donor. The universal acceptor is the blood type that can receive all other blood types, and that's type AB positive. The reason it's the universal acceptor is because it has A and it has B and it has the Rh protein, so it would be introduced to nothing new. Blood typing is based on the antigen-antibody reaction, that is, agglutination or clumping. You'll be doing blood typing in the laboratory. Here are 2 introductory videos that should be helpful. I hope you took the time to view the previous videos. They're very helpful. These are examples of blood typing results. In the center, you can see where there's no reaction on the left, and on the right, there is a reaction or agglutination or clumping. So if we apply that principle to blood types A and B, on the left-hand side, you can see that when it's given anti-A and anti-B and anti-Rh factor, that the blood type here is A positive. The positive is because it reacted to the anti-D or the Rh factor. On the right-hand side, you can see that A did not react, but B and the Rh factor did, so the blood type is B positive. Let's talk now about the Rh factor. The Rh factor is just another protein or antigen on the surface of some red blood cells. People who have this are said to be Rh positive. People who do not have this protein are said to be Rh negative. And we know that Rh positive is dominant over Rh negative. If we look at people's genotypes and a person has the genotype Rh positive, their blood type would be Rh positive, and the red blood cell antigen they have is the Rh antigen D. If their genotype is Rh positive, Rh negative, their blood type is Rh positive, and again the reason is Rh positive is dominant over Rh negative, and their red blood cell has the Rh antigen D. If the genotype is Rh negative, the blood type is obviously Rh negative, and they have no antigen or protein on the surface of their red blood cell. Although A, B, and the Rh factor are all due to the presence of a protein on the surface of the red blood cell, there's a big difference between blood type and Rh inheritance. Unlike the ABO blood group, a person is not born with the anti-Rh antibody. It requires prior exposure to produce the anti-Rh antibody. So, the first time a person is exposed, there's not a problem, but exposure to the Rh positive protein after this would be a problem if the person has been untreated. So what happens if you have a mother who's Rh negative carrying a baby who's Rh positive? Well, the risk is sensitization could occur. This means that if she ever carries another Rh positive child and is untreated, that her Rh antibodies would recognize the Rh proteins on the surface of the second baby's blood cells as foreign, and her antibodies would then pass through to the baby's bloodstream and attack those cells. So let's talk about when the Rh factor is a problem. The only time it can be a problem is if mom is Rh negative and dad is Rh positive and they have an Rh positive baby. So in the first case, mom is Rh positive, dad is Rh positive, the first baby is Rh negative, so mom's not being exposed to anything she doesn't already have. Baby 2 is Rh positive, but again, she has the same protein that mom has on the surface of the red blood cell. In the second case, mom is Rh positive, dad is Rh negative. First baby is Rh positive, second baby is Rh positive, but again, mom has that same factor on her red blood cell, so it's not a problem. In the third case, mom is Rh negative, dad is Rh positive. This could be a problem. Their first child is Rh positive, and if mom is untreated, then she would have antibodies to the Rh factor. However, the second baby is Rh negative, so she does not have exposure to that protein again, so there's no problem. In the fourth case, mom is Rh negative, dad is Rh negative, and both babies are Rh negative, so there is no problem. And the last case is the scenario of where you could have a problem. Mom is Rh negative, dad is Rh positive, and both babies are Rh positive. If mom was untreated after baby 1, then there could be a problem. One result of an Rh incompatibility is a condition known as erythroblastosis fetalis. In a mother who has had 2 children, both Rh positive, and she was untreated, this can occur in the second child. These are symptoms found in newborns with erythroblastosis fetalis. First of all, it is a hemolytic disease, and that means the red blood cells are destroyed or they rupture, and it's the result of an Rh incompatibility. The mother was Rh negative, the father was Rh positive. Both hemolysis and agglutination or clumping of the baby's blood will result. This leads to an enlargement of the liver and spleen. Because of agglutination and hemolysis, you expect this baby to be jaundiced and anemic at birth. The treatment for a newborn with erythroblastosis fetalis is a complete blood transfusion. Take a look at this short video to see how this is done. Obviously, it's going to be better to prevent the disease instead of trying to treat the disease. The prevention for this is a substance known as RhoGAM. RhoGAM contains anti-Rh antibodies that will remove the baby's antigen from the mother's blood. It's administered by an injection, and it's administered at about 28 weeks and within 72 hours after delivery. It's administered at 28 weeks because there may be future bleeds in the placenta, and it will last for about 12 weeks. Prior to this time, most tears would be too small to trigger an immune response in mom. Why 72 hours? Because mom's immune system begins to produce antibodies at this time. RhoGAM works by removing the baby's antigens from the mother's blood. It doesn't attack the baby's blood because it's too large to cross the placenta. The spleen removes these complexes from mom's blood. There are a number of different scenarios in which RhoGAM might be given to an Rh negative mom. After an abortion, after a miscarriage, following abdominal trauma, following an ectopic pregnancy, after an amniocentesis or CVS, which is chorionic villus sampling, or during or after a normal delivery.